or the structure of the membrane. So you have your phospholipids and cholesterol and glycolipids, you have your proteins, and then you have your carbohydrates that are the glycolipids and the glycoproteins that form the glycocalyx on the external side of the membrane. So let's take a look at membrane transport. The membrane, you remember, is selectively permeable. It can control what goes in or out of the cell. And there's three major methods by which things can move across the cell. There's simple diffusion, facilitated diffusion, and active transport. Simple diffusion and facilitated diffusion both rely upon a concentration gradient, and they move down the concentration gradient from high concentration to low concentration. And because of that, they do not require any energy input, no ATP. In contrast, active transport requires ATP because it's going to move a solute from low concentration to high concentration against that concentration gradient. And that's going to require an input of energy, usually in the form of ATP. So simple diffusion, by definition, is the movement of particles or solutes across the membrane from an area of high concentration to an area of low concentration. And the only things that can move across the membrane are things that are small and uncharged. So gases and lipids. So oxygen and carbon dioxide, here you can actually see carbon dioxide here. It diffuses from high concentration to low concentration across the membrane with no problems because itself is not particularly water soluble. Here you can see the concentration gradient for carbon dioxide. So lipids, steroids, some of the free fatty acids, those sorts of things can get in through simple diffusion. Okay, like oxygen, carbon dioxide. Everything else requires help. So that help is facilitated diffusion, and this requires the help of a protein. It doesn't require energy. Remember, it's still this idea of diffusion down the gradient, but it does require some help to get across that cell membrane. So those are the idea of the transport proteins. So we have pores, channels, and carriers. So pores are doors that are always open, right? So if there's a concentration gradient, if there's something is really high out here, and there's a pore for it, then it's going to move down its gradient. And water moves like this. So water moves through things called aquaporins. So water moves from high to low, or from high to low, depends on what's going on. And so we often say that water is freely permeable to the membrane because every cell has lots of aquaporins that enables water to move down its gradient. Channels, on the contrast, are doors, but they're doors that can be open or shut. Okay, we often say that they're gated. So we often say they're gated channels, and they can be gated by a lot of different things. They can be gated by light, they can be gated by chemicals, they can be gated by mechanical activity, so touch, by physical depression, by physical contact. They can be gated by electricity. They can be gated by lots of different chemicals, like things that you smell and things that you taste and things that are secreted by cells. So sometimes called ligand-gated channels. So if the stimulus is present and it binds to the channel, that channel can open and let in something. Now, whatever it lets in, right, is always going to move down its gradient. So in this case, we have a ligand-gated sodium channel. So ligand is just another name for a chemical that activates a channel, right? And so this channel, which is a receptor for this ligand, it receives information that ligand's present. When the ligand's present, the receptor opens and sodium moves down its gradient. If this was a ligand-gated potassium channel, then a ligand would bind, and instead of sodium moving, it would move potassium. And our channels are very specific, and in their name, it always tells us what they do. And they're usually ion channels. And then we have carrier proteins. Carrier proteins are proteins that actually have to grab a hold of the solute that it's moving, and so they take a little bit more time to work. They're not they can get saturated, and we often look at carrier proteins for things like amino acids and carbohydrates and nucleotides. So ions usually move by channels, and then the amino acids and nucleotides usually move by carrier proteins. And we'll talk more about carrier proteins when we start talking about absorption of nutrients. So osmosis, it's worth our time to take a minute to talk about osmosis. Osmosis is the movement of water through a selectively permeable membrane. Prior to this, we've been talking about the movement of solutes, sodium, ions, nucleotides, amino acids, gases, lipids. Here we're going to talk about the movement of water. You do need to know the definition of osmosis. Osmosis is the movement of water, it's always the movement of water. And it's the movement of water from an area of more water to an area of less water. So here you can see in this picture that there's lots of water here and relatively littler here. So water is going to move across the membrane down its gradient. But you'll also notice you can define it as it's the movement of water from an area of fewer solutes to an area of more solutes. So note that on side B that there are no solutes, and on side A there's lots of solutes. So water moves from where there's few solutes to where there's lots of solutes. And that's going to actually drive the level of this fluid up, assuming those solutes cannot move. So osmosis, this is why your grocer puts sprays your food, right? Sprays the produce, because it makes the fruit and the vegetables look more plump because they're spraying it with pure water, and that water moves into the cells and makes them look plumper. And again, we've already talked about aquaporins. Aquaporins are channel proteins that allow water to freely move in or out of the cell. And it's determined by that tonicity. And you remember we discussed tonicity when we were looking at homeostasis. So active transport is also protein mediated, but it's active and it moves the solute against its gradient. So it moves it from low to high. And energy, usually in the form of ATP, is required. The best example of that is the sodium-potassium pump. And we're going to come back to the sodium-potassium pump a number of times. So let's talk about it here. The sodium-potassium pump is a protein that pumps 3 sodiums out. Sodium is very high outside the cell, but we're going to pump even more of it out. Okay, at the same time, so this is kind of a co-transporter, it brings in 2 potassiums. And potassium is really high inside the cell. That's just the natural state of living cells. That sodium's high on the outside and potassium's high on the inside. So the sodium-potassium pump, or the sodium-potassium ATPase, consumes ATP, consumes energy, and in doing so, it moves 3 sodiums out and 2 potassiums in. And the importance of this we'll talk about when we talk about action potentials in muscles and in neurons. So the sodium-potassium pump is a great example of active transport. It does help us to talk just briefly about primary versus secondary active transport. Primary active transport relies upon ATP. So that sodium-potassium pump we just talked about uses ATP to do the pump. That's primary active transport. But another method by which we can consume energy, we can use energy, is if we take something that's high outside the cell and we let it come in, and we take along something that's low outside the cell. So in this case, sodium is moving down its gradient, but glucose is moving up its gradient. And this is sometimes called secondary active transport, or ATP independent. And it uses the energy from the ion gradients in sodium. And we're going to talk a lot about this type of transport in Bio 202. So you don't really need to worry too much about it, but it's a good idea to kind of start wrapping your head that energy can come from a couple of different things, either from ATP or from a concentration gradient. We can actually use a concentration gradient to do work. And you can use an analogy of this as a sawmill, where it uses water to turn a water wheel that then makes electricity that can then, say, cut wood or grind flour or what have you. So the other method by which we can let things in and out is called bulk or vesicular transport. And we can either get things out or we can get things in. So exocytosis is the process by which we secrete things out of the cell. So this is going to be things that are bulk, lots of it, where 1 or 2 isn't enough, so we need to release hundreds. And so exocytosis, we're going to package things in vesicles. That's why it's also called vesicular transport. And those vesicles fuse with the membrane and release their contents. Similarly, we can bring things in. So here we have our endocytosis. Endo is bringing things into the cell. Here we're capturing things in this little pit. We're going to pinch off that pit, and then we have our endocytosis. And here you can see a picture of that as well, where we bring things from outside in. A specialized case of that is called phagocytosis. White blood cells rely upon that to engulf really large particles to get them out of the environment, things like bacteria and debris, to get them out of the environment. So that's phagocytosis, and it's a specialized form of endocytosis. Now, receptor proteins are the other major protein component of the cell, and they receive information about the environment. And when they receive that information, they change their shape. And they change their shape, that initiates a cellular response. We're going to talk a lot about what the cellular responses can be, but just in a brief list, they can be metabolic, meaning they can turn on or turn off activities. They can be mitotic, which means they can cause the cell to divide. They can cause the cell to commit suicide or to kill itself for the good of the whole organism. They can cause cells to grow. They can cause cells to make things, for instance, to make hormones or to make more proteins or sugars. And they can cause the cell to develop or differentiate and specialize into particular types of cells that the body might need. So receptors we're going to talk a lot about. And then pretty much anything you could think about a cell doing, the receptor probably can tell it to do so. But only when the ligand or the agonist is outside. So only when told to do so. So receptors are there to enable you to see, the cells to see what's going on around it and to respond appropriately. So that's the cell membrane and the membrane functions, and we're going to come back to this. So if you have any questions, please do email me or contact me, because if this is kind of a loss for you, then it's going to make a lot of what we're going to talk about throughout all of the systems very, very difficult to understand. So please take a look at this information, read your textbook, do your review questions, your assignments, and then email me if you have any questions.